Dr. Nardi here, back with another video. Today we are going to talk about the world of comparative neuroscience. Now, if you've been following the channel, you know we often talk about the human brain. But to truly understand our brains, we actually have to look at the brains of other species. Comparative neuroscience is the study of human and non-human animals to better understand brain structure, function, and evolution. It helps us answer two fundamental questions. First, what are the basic principles of biological organization that apply to all of us? And second, what makes the human brain distinct? In this lecture, we are going to cover how we choose model systems, the controversies in comparing different brain structures, how brain size relates to intelligence, and the cutting-edge technology of brain organoids. Let's start with a practical question. How do scientists choose which animal to study? We call these model systems. Animals chosen with the expectation that what we learn from them will apply broadly to other species. Historically, these choices were often driven by very practical physical traits. For example, consider the squid. A lot of what we know about action potentials, the electrical language of the brain, comes from work by Hodgkin and Huxley on squids. Why? Not because squids are incredibly smart, but because they have massive axons that were large enough to easily study in a lab. Similarly, Hubel and Wiesel won a Nobel Prize for their work on cats. They chose cats because they are visual hunters with acute vision, making them perfect for understanding how the brain processes sight. Today, the most common model system is the mouse. They are small, cost-effective, breed quickly, and are genetically similar enough to humans to be incredibly useful for biomedical research. But sometimes we choose an animal because it has a superpower or a unique adaptation. We study bats to understand echolocation and multisensory integration. We study naked mole rats or voles to understand specific social behaviors or longevity. Now, I want to be clear about the ethics here. Research involving animals is heavily regulated. In the United States, we have committees called IACUC, Institutional Animal Care and Use Committee. Before any study begins, researchers must prove that the study is justified, that the number of animals is minimized, and that strict measures are taken to minimize pain. It is a rigorous process designed to ensure humane treatment. Once we have our model system, how do we compare it to a human? This brings us to the concept of homology. Homology refers to structures that evolve from a shared ancestor. Think of a human hand and a bat's wing. They look different and do different things, but they are homologous because they evolved from the same limb in a common ancestor. In the brain, this gets tricky. Let's look at one of the biggest debates in the field: bird brains versus mammal brains. If you look at a mammal's cortex, it is organized into distinct layers, specifically 6 layers of gray matter. But if you look at a bird's brain, it doesn't have layers. Its neurons are clustered into groups called nuclei. So which part of the bird brain equals the human cortex? There are two opposing theories here, and they depend on what scale you look at. First, we have the isocortex DVR hypothesis. This looks at connectivity, the macro scale. Scientists noticed that a part of the bird brain called the DVR receives sensory input just like our cortex does. Because they connect to the rest of the brain in the same way, proponents argue they are homologous. But then there is the claustroamygdala DVR hypothesis. This looks at development, the micro scale. When scientists looked at gene expression in the embryo, they found that the cells destined to become the bird DVR look a lot like the cells that become the amygdala in mammals. So is the bird DVR a cortex because it acts like one, or is it an amygdala because it is built like one? This debate highlights how difficult it is to compare brains across distantly related species. Now let's talk about size. There is a general rule in nature called allometry. This means that as an animal's body gets bigger, its brain gets bigger, and specific parts, like the cortex, scale up in a predictable way. However, we sometimes see what we call a grade shift. This is when a brain region is larger or smaller than the math predicts. Primates, for example, have a grade shift regarding the cortex. We have much more cortex than a rodent of the same size would have. We also see specializations based on lifestyle. This is a use it or lose it evolutionary scenario. Take the platypus. It hunts underwater with its bill, detecting electrical impulses and touch. If you look at its brain, the somatosensory cortex, the touch area, is massive. Conversely, look at the blind mole rat. It lives underground. Its visual cortex is tiny, but its touch areas are huge. This tells us that brain real estate is expensive. Evolution invests energy only in the sensory systems the animal actually uses. So we know about the size of these regions, but how do we know how they connect? In animals, we use track tracers. This is the gold standard. We inject a dye into the brain, and it physically travels down the axons, the nerve fibers. Anterograde tracers move forward from the cell body to the terminal, while retrograde tracers move backward. This lets us draw precise physical maps of brain wiring, but we cannot inject these dyes into living human brains. So, for humans, we rely on neuroimaging, specifically diffusion MRI. This tracks the movement of water molecules. Water diffuses more easily along a bundle of axons than across them. By tracking water, we can digitally reconstruct the highways of the brain. It's not perfect. It's a reconstruction, not a direct picture, but it has revealed massive insights. For example, the arcuate fasciculus is a bundle of fibers connecting the frontal and temporal lobes. In humans, this pathway is massive compared to macaques or chimps, which suggests it is crucial for our unique language abilities. We can also use animals to understand complex behaviors, like love and social bonding. My favorite example involves voles. We have prairie voles, which are monogamous and form lifelong pair bonds. Then we have meadow voles, which are promiscuous and do not pair bond. They look almost identical. So what is the difference in their brains? It comes down to a receptor for a hormone called vasopressin. Prairie voles have high densities of these receptors in a reward center of the brain called the ventral pallidum. Meadow voles don't. Here is the amazing part. Scientists used a virus to genetically increase these receptors in the promiscuous meadow voles. The result? The meadow voles started forming pair bonds. This is profound because it links complex social behavior to specific molecular changes. And interestingly, variation in this same receptor gene in humans has been linked to how men bond with their partners. Finally, I want to talk about the future of this field. How do we study the differences between human and chimpanzee brain development without harming them? We are now using brain organoids. We can take skin cells, reprogram them into stem cells, and grow them in a spinning bioreactor. They self-assemble into tiny three-dimensional spheres that mimic the layers and cell types of a developing brain. We can use RNA sequencing to read the genetic activity of these organoids. When scientists compared human organoids to chimp organoids, they found something fascinating about our timing. Human organoids mature much, much slower. This is called protracted development. While chimp neurons matured quickly, human neurons stayed in a young, plastic state for longer. This suggests that the secret to human intelligence might not just be that our brains are big, but that they stay unfinished for longer, allowing us to learn from our environment for years during childhood. To wrap up, comparative neuroscience isn't just about cataloging animal facts. Whether we are looking at the giant axon of a squid to understand electricity, the visual system of a cat, the social life of a vole, or a cluster of stem cells in a petri dish, every model system provides a piece of the puzzle. By combining these pieces, we get a clearer picture of the biological rules that govern all life and the unique evolutionary changes that make us human. That's all for today's lecture. Make sure you review the differences between the isocortex and claustroamygdala hypotheses, and I will see you in the next video.